Hello, everybody. Hello, everybody, and a warm welcome to the opening presentation in the inaugural LabRoots Precision Medicine virtual event. I'm Kevin Davis, the publisher of CNEN, Chemical and Engineering News for the American Chemical Society. We are delighted to be the official media partner and co-organizers of this groundbreaking virtual conference hosted by LabRoots. LabRoots is the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Our opening speaker is Ruth March from AstraZeneca in the UK. For scheduling reasons, we're unable to offer a live recording today. We're pre-recording this a couple of days early, and for quality reasons, we're just going to rely on an audio presentation, so you don't have the undoubted pleasure of either seeing me or Ruth on video. So we apologize for that. But let's get going with a couple of housekeeping notes, and then I will introduce Ruth for our opening presentation. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. And if you encounter any technical problems during this or any of the other talks, either viewing or hearing the presentation, please click on the support button, which you'll find top right of your screen, or you can submit the problem through the green Q&A button, which you see on the lower left-hand side. As this is an educational webinar, we're pleased to be offering continuing education credits. At the conclusion of the presentation, if you click on the CE button, which you'll also find in the lower left-hand corner of the web page, then follow the instructions to apply and obtain your credits. So, with the honor of batting first in today's Precision Medicine virtual event, it gives me pleasure to welcome all the way from Cambridge, England, Ruth March, who is Vice President and Head of Personalised Healthcare and Biomarkers at AstraZeneca. We're delighted to get the perspective of a leading big pharma representative as we hear about a number of different views and strategies and projects in this relatively new aspect of precision medicine. Ruth leads AstraZeneca's team of diagnostic and biomarker experts delivering companion diagnostics. She's driven adoption of personalized healthcare approaches by something like 70% of AstraZeneca's pipeline, partnering with many diagnostic companies in the process. She has a long and distinguished academic background, having worked as a researcher and professor in immunology and genetics research at the Universities of London and Oxford before joining AstraZeneca. The title of her opening presentation here today is How Personalized Healthcare is Transforming Drug Development. Ruth, a very warm welcome to you.
Thank you, Kevin, and thank you particularly for that warm introduction. And I'm delighted to be speaking to you here today by the wonders of modern technology, reaching out all across the globe to the community of experts who are interested in this fantastic topic of precision medicine, or as we call it in AstraZeneca, personalized healthcare. So what is personalized healthcare, or indeed precision medicine? Well, there are many terms, and my experience is that if you get 10 experts on this subject together in a room, you will end up with at least 11 definitions. So we prefer to start by defining it as to how we see personalized healthcare or precision medicine delivering. And it's all about delivering the right treatment to the right patient. It recognizes that successful treatment is about more than the drug alone. It can involve a lot of extra things. In AstraZeneca, we particularly focus on the role of companion diagnostics and other biomarkers to select patients for the right treatment and optimize treatment. It's obvious that this is not an easy thing to do, but when we do it, and we do it successfully, it ends in better outcomes for patients. So our approach to personalised healthcare is centred on the patient. It's integral to our business and to our values of following the science and putting patients first. As I said before, our aim is to improve patient outcomes. And when we really do that, when we deliver a step change in clinical benefits, that helps us to deliver value to payers and to the business. One of the things that I'll be talking about later is how we apply personalised healthcare across all of our core therapy areas and how we integrate it from the very start of R&D. Now, AstraZeneca, unlike some companies, does not have a diagnostic business. We don't have a diagnostic arm. So that gives us the freedom to work with diagnostic partners all across the globe, and we work in a particular way to select those partners who are best for the drug and for the disease of choice. Now, as Kevin said earlier, we have been working in this area for about 10 years, and we have reached the stage now where more than 80% of our clinical pipeline have a personalised healthcare approach. These are drugs that are either developing with companion diagnostics or they are actively involved in research to go down that approach. You probably can't see the detail of this slide. I just want to pick out a few things for you. So first of all, those projects with the green dot, that means that they have that personalised healthcare approach, are seen across every phase of drug development. They're also seen more or less equally in small and large molecules. And as we said before, you can see examples in every therapy area and not just in oncology. As we work with diagnostic companies and other leading academic centres, we are creating a leading position with those strategic partnerships. We have some of these examples down here on this slide, and again you can see how they are spread throughout the globe. So why is this so important? You'll all know that the biopharmaceutical research and development process is one that starts off with a lot of medicines. Potential candidates that go into the start of clinical trials and even in preclinical development then narrow off through clinical development so that only one FDA-approved medicine comes out at the end. In AstraZeneca, we believe that the solution to this problem lies in quality and not just in quantity. And we have published a paper on what we call the 5 R's. And these depend on having the right target, the right tissue, the right safety, and the right commercial potential for our drugs. A central part of this is selecting the right patients for the testing of our drugs. So let's take a look at one example of where we've done this. Now, many of you will know that the first EGFR inhibitors, drugs like gefapixant, highlighted the importance of identifying the right patients. But in the case of these early targeted therapies, those biomarkers were not discovered until some years post-launch. I believe for gefapixant it actually took 6 years post-launch to identify the biomarker EGFR mutation that is shown on this slide. And then the emerging clinical development showed that resistance to those first-generation drugs was correlated with the appearance of secondary mutations in the EGFR gene. And these were the T790M mutations. And you can see here with the separation between those curves with patients that were negative for that mutation, so for having significantly longer survival than those that carried them. So what we wanted to do was to take that information from the clinic, from the patients, and feed it back into drug discovery to generate the next generation of EGFR inhibitors. So this diagram starts to show you the fantastic work that was done by the chemists and the drug designers here at AstraZeneca to actually design a new drug that would be able to inhibit not only the EGFR mutation, but also this resistance mutation of T790M. So you can see here that 66% of acquired resistance is due to that T790M mutation in exon 20 of EGFR, and it's so-called the gatekeeper mutation. So we then developed a new drug, and its code name was AZD9291, which was designed to target that resistance mutation as well as the original mutations in EGFR. And as I flick through here, you'll be able to see how that yellow portion in the crystal structure at the top right-hand corner of the structure at EGFR shows how the original T790M blocked the binding of the first generation medicines, gefitinib and Talceva. So the new structure of AZD9291 actually is able to bind in a different place. It avoids being blocked by that T790M mutation and is still able to inhibit the action and the activity of that mutated receptor. So that was a fantastic piece of drug design, going into very early drug discovery, even before the drug went into the clinic. And once we'd done that, once we learned from the clinic, we'd understood the biology, and we were able to identify the right patient population from the very start of clinical trials. This is what happened. This remarkable curve shows you the overall response rate in very early clinical trials, and all of the bars there represent the response from an individual patient. Anything that is above the black line at 0 is an increase in tumour size, and everything below that line is a decrease in tumour size. And the cutoffs there show you the best percentage change from that target lesion. So this is a graph, believe me, that you don't see very often in drug development. And it emphasizes the importance not only of selecting the right patients, but taking those clinical messages back into the start of drug discovery and designing the next generation of drugs. The result of all of that was, I'm delighted to say that AZD9291, now known as Tagrisso, was approved by the FDA in November, and this was one of the fastest clinical development programs ever known to bring that new and innovative medicine to patients. And just recently, in fact, on the 3rd of February, the drug was also approved in the EU. It's a first-in-class treatment for patients with that EGFR T790M mutation-positive non-small cell lung cancer. The remarkable thing about both of these approvals is that they were both linked to the approval of a companion diagnostic test that was developed at the same time as the drug and delivered on the same day. So in the case of the FDA, that was a PMA approval for a companion diagnostic. You can go and look this up on the FDA website. That was approved on the very same day as the drug. In the EU, it's even better because the approval for the drug coincided with the launch of a diagnostic test that is approved both from the tumour sample and from plasma. So for patients that don't have that tumour sample available, they can be diagnosed and have Tagrisso as a treatment option from a plasma test. We'll go on to talk a bit more about that in a minute. So what are the new horizons in diagnostic development and precision medicine? We are proud to be joining with our partners in the evolution of the next generation of diagnostic technologies. We're seeing a very exciting move from single markers through to multiplex testing. We're moving from very invasive procedures to those that are minimally invasive, and from a semi-quantitative approach to pathology to digital pathology. The future, we believe, will be to move more of that diagnostic testing earlier in the patient journey. So preferably, patients would only have to get tested once at diagnosis, and they would have a comprehensive panel testing that would then be integrated with their clinical data. And of course, this would be happening across all therapy areas. So let's come back to the role of circulating tumour DNA in plasma and how we believe this is putting patients first. We started to work on this technology back in 2006 when it was something that was being explored by research laboratories, but some people thought it would never make it to the stage of being a companion diagnostic that could be used to guide therapy in the clinic. So we sponsored some of this research, and we now have the world's first ctDNA-based companion diagnostic. As I said before, about 25% of lung cancer patients do not have solid tumour samples available. That can be because the tumour sample was used up during the pathology that led to their diagnosis, or it could be because that sample is just not of high quality enough to do molecular analysis. Before this diagnostic was approved, those patients had to undergo a very invasive and expensive procedure. Now they can use this plasma test, which just comes from taking a normal blood sample. And if they are positive by that test, they can be offered one of our targeted therapies as a treatment option. We're also making great use of next-generation sequencing, which we believe will transform medicine over the next few years. Now, next-generation sequencing, and we're using a technology, incidentally, that was invented in Cambridge, not far from where I am now, is up to 1 million times faster and more sensitive than previous sequencing technology. So we are actually using NGS in clinical trials to select patients for treatment and also to develop the gene panels as companion diagnostics. In particular, NGS enables us to conduct so-called basket trials. The idea of these is that patients are only tested once. They come into the clinic and they are tested with a comprehensive gene panel. And depending on their results, they can then be assigned to the best treatment for them. This helps us to speed up clinical development, and we also believe that it benefits patients. We're also very excited about the use of NGS in research. So as we look to advance personalised healthcare and bring its benefits to patients in all our therapy areas, we are looking at many different techniques. So I've talked about the circulating tumour-based DNA in oncology. And in respiratory disease, we're looking at novel imaging techniques that can look at how drugs are taken up into the lungs and measure receptor occupancy. In neuroscience and Alzheimer's disease, the use of imaging and PET ligands is much more well advanced, and we do have some trials going on at the moment where that is used to help select patients for treatment. In asthma, we're looking at protein markers and cellular assays. And finally, we're using genomics to reveal drivers of disease in CVMD. That's cardiovascular and metabolic disease. So all of this wonderful science is very exciting to be involved in. But what we really want to do is to bring people together to deliver what we're talking about, to deliver that promise of personalised healthcare to patients. And this is where we rely on the linkage of that drug and the diagnostic going together. Now, what we've learned over the years is that delivery to patients takes many partners working together. It can take all aspects of the healthcare system because we're involving many more people than the conventional routes of drug delivery. Some of the key components there are putting together our scientific understanding, our diagnostic platform, and regulatory acceptance in all of the different countries that we work all over the globe. Putting it into practice means demonstrating that value that we bring to patients and physicians. And obviously, the actual assays and the lab infrastructure have to be available at the point where the physician is practicing and the patients are waiting for therapy. And finally, as all of that comes together, the new way of working can be recognized in clinical guidelines and practice. So today, we are looking at a world where there are more personalized healthcare products than ever before. Just looking at the FDA website, where companion diagnostics are listed, is really encouraging in the way that the speed of those developments are coming together. So what will be the future for personalized healthcare or precision medicine? Could the patient of the future be somebody like this? Instead of sitting in a doctor's office with all other patients, sick patients, all together, it could be somebody that's maybe gone to their local pharmacy or even their local supermarket to donate a sample and have that sample tested. The results of that test could be making their way by wireless to the patient's family physician, so that the next time that they call in, the best therapy for them will be already dispensed and waiting for them. Who knows? And finally, I just want to highlight to you that AstraZeneca realizes that we cannot achieve any of this on our own. So we have a website that is called Open Innovation, and I encourage you to dial into that and see the opportunities that we have to advance research together. And with that, I'll hand back to Kevin.
Thank you so much, Ruth. What a fabulous opening to our inaugural Precision Medicine virtual event. A wonderful perspective and some really exciting data included in there. So yes, I will follow up with you with a few questions. I very much liked your opening quip about if you assemble 10 experts in a room to define precision medicine or personalized medicine, you'll end up with 11 definitions. But I wonder what has been your personal or your company's sort of response or reaction or reception to the precision medicine initiative announced from in the Obama State of the Union just over a year ago now, and I wonder how you would react, and whether you feel that's going to lead to some useful innovation and further funding in this field.
Thank you, Kevin. Yes, we are very excited about the Precision Medicine Initiative, and we don't even mind whether it's called Personalised Healthcare, Personalised Medicine, or Precision Medicine. Any initiative that is aimed at exploring science, driving that linkage between tests and biomarker data and therapy forward really has our support. So we have an initiative in the UK called Genomics England, where we are an active member of the Gene Consortium, and we also have in the US the PMI, or Precision Medicine Initiative, which we also definitely support. I would love to see these initiatives really gaining more data about advancing healthcare. There are many basic science questions where we need many more numbers of patients really to volunteer and become part of getting all of this healthcare data together. What we've realized is that, particularly in common medicines, some of the effects that we're looking at are probably quite rare and quite subtle. So that's why we need these big initiatives to actually advance the science, and that's really where we would want to support these type of initiatives, and indeed want to see many more of them.
Ruth, you just interestingly mentioned Genomics England, and by coincidence, our very next speaker here at the Precision Medicine virtual event is Tim Hubbard, the director of Genomics England. Could you say just a little bit more about what is the nature of your collaboration between AstraZeneca and Genomics England, which, as most people know and will hear in more detail at the top of the hour, is building a large biobank of I believe, some 100,000 individuals? Yes,
thank you. So our involvement in Genomics England was, I guess we were one of the first companies interested with our colleagues from elsewhere in the pharma industry in the UK, and then more generally including companies in the US and globally in this exciting initiative. And we were extremely pleased when the UK government sponsored this approach to genomics research. So as you said, Kevin, the exciting thing about it is that for the first time they have the largest genomics public-private initiative in the world to sequence and to do whole genome sequences of 100,000 genomes. And many of those are from cancer and from rare diseases. We have interest in any number of fields connected with these. First of all, there's the actual discoveries that I am sure the scientists and the expert scientists in Genomics England will make. Secondly, there's actually how you do a research initiative like this within the National Health Service. So there's many aspects of this, from consenting patients to educating physicians to actually getting the interpretation and using the data within the National Health Service, which I think are tackling some of the toughest questions within actually applying personalised healthcare so that it does deliver to patients.
Excellent, thank you. Let me just quickly ask a couple of questions about Tagrisso, this tremendous success story that you've led for forms of lung cancer, targeting forms of EGF receptor that have been mutated. You mentioned basket trials, Ruth. Was that approach part of the trials that has sped the development of Tagrisso, or is that something that's come along since you completed those clinical trials?
So the basket trials we're taking part in we have one in the US which is called LungMap, and actually Tagrisso was not included in that. There were some other drugs from our pipeline that have been included in that, but it's the same general approach where patients were actually tested, they were sequenced, and then depending on the approach, depending on the results of that sequencing, they would be placed either on one of our drugs or indeed on drugs from other companies. So it was a true collaboration. That's still going on at the moment. And the other collaboration that we're involved with there is in the UK with the Stratified Medicines Initiative. And there the connection is through Cancer Research UK, and it's a similar proposition, except that many more of our early stage drugs are included in that panel where patients can be tested and then placed onto very early stage trials. And again, it's not just our medicines that are involved with that, but some, I believe, from other pharmaceutical companies as well. So both very innovative ways of doing things, and ones which I think are obviously good for patients because they have that one testing experience and then they can be assigned the best treatment for them. But it will be interesting how the regulators then respond to how you take the results from those early clinical trials and take them forward.
Yes, and my final question, Ruth, is you mentioned your interest and work in next generation sequencing, your partnerships. There's a technology, of course, that began at Cambridge with Solexa and then became Illumina. And of course, Illumina made a lot of news at the beginning of the year with the spin-off of a liquid biopsy company called Grail, and there are many other companies also exploring this. I'm just curious whether, given the importance that you've highlighted so nicely in your presentation regarding companion diagnostics, this must be a field that you're looking at pretty avidly, I would imagine.
Yes, so our collaboration with our partnership with Illumina is centered around, which was announced, I believe, last year, is centered around the development of lung panel tests that then can be used in developing many drugs within our pipeline. So it's not yet directly connected to that other announcement that came out in January of this year, but it's certainly, it's still, again, it's a challenging area, not just technically and scientifically, but very much through the regulatory part. So all this development of companion diagnostics is something that goes through the FDA, the EMA, and other regulatory bodies. And this is not always as straightforward as it might seem once you have a panel of maybe tens of genes on the same test.
Well, Ruth, thank you very much indeed. And again, I apologize to the audience that we were unable to see you on video for the opening presentation today, but I trust that didn't detract too much, and that the information you've been able to provide has really supplied a tremendous kickoff to our Precision Medicine virtual event. That was Ruth March from AstraZeneca, where she is Vice President and Head of Personalised Healthcare and Biomarkers for the pharma company, giving a wonderful overview to kick things off. And at the top of the hour, our next set of presentations, we hope you'll stay tuned for those. You'll hear from Tim Hubbard, the Director of Genomics England, just mentioned in Ruth's presentation, and there'll also be a sponsored presentation from QIAGEN. So you have a choice, and we hope that you'll, if you can't stick around for the whole series of events running throughout the day here at the virtual conference, that you'll at least come back for a few more. And this and all of the presentations will be available on demand for viewing at your convenience and your colleagues for the next 6 months. Through August 2016. You'll receive an email from LabRoots after the event telling you when this webcast and others will be available for on-demand viewing. So that's all for our opening presentation. We hope now that you'll check out the exhibit hall and many of the other features and resources that are available. I particularly want you to spend time and talk to some of the exhibitors who have made this inaugural Precision Medicine virtual event possible. So for now, I just want to thank LabRoots and Ruth March in particular for joining us, and we'll see you again for another presentation, hopefully at the top of the hour. So this is Kevin Davis. Thanks for joining us, and bye-bye for now.